thyroid function. And research interests include the role of nuclear hormone receptors in human disease, genetic and acquired disorders of the hypothalamic pituitary thyroid axis, novel approaches to sparing hypothalamic pituitary function in patients with cellular and paracellular tumors, and functional imaging in endocrine neoplasia.
Well, thank you very much, and it just remains to say it's a great pleasure to be back again in India. Thank you for the invitation. So what we're going to do in the next 25 minutes or so is use some lessons from nature just to remind ourselves a little bit about thyroid physiology. And I think it's actually very important that when we treat common thyroid diseases, we think back to the mechanisms of how thyroid hormone action is regulated, how it's produced, and how it's metabolized, to actually understand some of the consequences of what we see. So in Cambridge for some considerable time we've had a big interest in looking at patterns of thyroid function tests. Now many of these will be very familiar to people because they're the standard patterns, they're the normal thyroid function, or indeed those that are very traditionally recognized as reflective of overt thyrotoxicosis or indeed hypothyroidism. And of course there are many conditions which can cause these, and you'll be very familiar with many of those. However, we've had a big interest in looking at patients who have what you might call atypical or slightly unusual thyroid function tests. For example, where the thyroid hormone levels are normal but the TSH is low or raised, or in patients where the free thyroid hormone levels are low or indeed raised but the TSH is inappropriate. And there are many, many different causes of each of these patterns as well, and all of this information is readily available in these publications, which are all freely available. And if you actually start to study some of these conditions, you start to understand much more about thyroid hormone regulation, action and metabolism through looking at what sometimes are considered to be relatively rare conditions, but which are actually more common than often recognized. So I need to remind you that this is a very classical endocrine system. It's a very typical feedback loop. TRH driving pituitary production of TSH, of course producing T3 and T4 from the gland in a ratio of T4 to T3 of about 14-15 to 1, and a very traditional negative feedback loop. And of course thyroid hormone is critical for everything. Without thyroid hormone during development, you're in big trouble, and indeed without thyroid hormone during childhood and in adulthood, you're likely to have many, many problems. Now we need to remember that when thyroid hormone gets to those various tissues, this is a real masterclass of how to regulate tightly the action of a hormone, because there's lots of different factors which determine whether thyroid hormone actually at the end of the day has an effect. First off, for a long time we used to think that thyroid hormones used to diffuse into cells, and we now know that they're actively transported inside. Thyroid hormone has to be converted. One iodine difference between T4 and T3, and yet T4 is inactive. T3 is the biologically active hormone, and then once T3 actually finds its way inside the nucleus, it interacts with a whole family of different regulatory proteins to direct transcription. So this is an extremely well-coordinated but very protective effect for the cells, because it's only when you actually get T3 into the nucleus and interacting with its receptor that you start to see the effects of thyroid hormone. So in terms of some examples, we're actually going to think or start actually within the central regulatory area, which is actually looking at pituitary TSH production and looking at some examples of how we can gain insights into what it is that controls TSH production that then of course drives the gland. So I'm going to begin with a couple of case vignettes. These are two small children. The first one was actually a 3-week-old male who was referred having developed poor feeding, dry skin and constipation. And I think you'll all agree that this is a pretty undesirable set of thyroid function tests to have. The TSH is very low, in fact almost undetectable by this assay, whereas the free T4 is also low. Now this child had otherwise normal pituitary function and the MRI was unremarkable, so this is isolated central hypothyroidism. And lo and behold, at pretty much the same time we get to see this child. This is a 3-week-old female, this time coming to attention because of prolonged neonatal jaundice, but a not too dissimilar pattern of results apart from the fact that here the TSH is measurable, but although measurable, it's clearly very inappropriate for a child with a low free T4 level. And again, normal pituitary function and an MR which is pretty unremarkable apart from an incidental Rathke's cleft cyst. So these are two children presenting with a very similar problem but with very different causes. So we need to just remind ourselves a little bit about the biology of how TSH is produced. So of course in the thyrotroph, TSH is generated. It comprises two subunits, that common alpha subunit and then the beta specific subunit. And that common alpha subunit, of course, is required for the production of other hormones such as LH and FSH and also for HCG. But there are some other shared activities going on here in these cells, because if we think about lactotrophs, TRH, which is the trophic factor for driving thyrotrophs, is actually also an important factor for driving prolactin production from lactotrophs. And we need to bear in mind this crossover in terms of entities such as the alpha subunit or key regulation through the TRH signaling pathway to start to understand the differences in these children. So, whereas primary congenital hypothyroidism has got about a 1 in 3,000 live birth incidence, central is rarer, about 1 in 40,000. So if we go back to this first child, a TRH test revealed an absent TSH response but a normal prolactin response. And that gives us a really important clue to working out where the problem is. Because here, we're unable to assemble TSH, but we're making prolactin reasonably. So that implies that this is not an issue in relation to how TRH is signaling. It tells us that it's much more likely to be an issue in relation to how TSH itself is put together. And given that the LH and FSH are normal in this child, it's unlikely to be an issue with the alpha subunit. And in fact, just through simple deduction alone, you can work out that this is likely to be a problem unique to the beta subunit. And in fact, this is one of the children of an increasing group now recognized to harbor mutations in the beta subunit of TSH. And if you have a loss of function mutation or a truncation in that beta subunit, you simply cannot form the dimer of the alpha and TSH beta, so you do not get any mature TSH. Similarly, you therefore cannot detect that TSH in the assay. So this is biologically inactive and immunologically inactive TSH. So this child has got isolated TSH deficiency. This is quite different. If we progress. This is quite different to the second child. Here, when we performed a TRH test, we saw that there was an absence in the TSH response and also the prolactin response. So coming back to our model again, here we're now looking at a defect potentially at this level because we're neither making TSH or prolactin, and that's exactly what we see. And this is one of the children who has a homozygous mutation, recessive, within the TRH receptor. So this is defective signaling of TRH due to loss of function of the TRH receptor. So here are two children presenting in an almost identical fashion, and yet they've got two different molecular mechanisms underpinning their disease. And then here comes a third child, another child of 3 weeks of age, this time with a similar presentation, with similar biochemistry, but on this occasion, when we do the TRH test, when we're looking at prolactin and other parts of pituitary function, we find the basal prolactin is low, but the pituitary function is otherwise normal. And so for all intents and purposes, we start to wonder again whether this is a defect in TRH signaling, because TSH is low inappropriately, and the prolactin is not as it should be. However, when we sequence the TRH receptor gene in this child, it's completely normal. And this is the excitement of dealing with these sorts of cases, because up to this point, the story would have been, well, this had to be a TRH or a TRH receptor issue. But it's not. So what is it? And it turns out that we learn more about the regulation by identifying mutations in a completely novel gene in relation to central thyroid disease. And this is IGSF1, immunoglobulin superfamily-like protein 1, which it now turns out is a key regulator of thyrotroph function. It's a membrane glycoprotein. We still don't fully understand its roles. It's X-linked disorder. We do know that there are now many, many people who are potential affected by this condition. And what's interesting is that although X-linked, it's not just the males who can present with central hypothyroidism. And as I say, this is a membrane-bound protein, and for example, when we studied the very first mutation here, we could see that the protein itself does not actually make it out onto the membrane compared to its normal wild-type counterpart in the center there, unless you permeabilize the cells so that you can actually then gain access to the protein inside. So the protein is being made, but it's actually contained within the cell and not making it to the membrane. And this is an interesting disorder because it turns out that central hypothyroidism is just one part of the phenotype of IGSF1 deficiency. IGSF1 is also very strongly expressed within the gonads, and so another key part of the phenotype is macroorchidism, which is a prominent finding in people with IGSF1 mutations. And what is also striking is the age at diagnosis. We have just recently diagnosed individuals who are presenting for the first time in life, despite having had central hypothyroidism, with a free T4 level of around 8 picomoles per litre for the first time ever at the age of 70, 75. Now that's a bit of a challenge, because if they've lived their entire life with a free T4 of 8, do we really want to correct it? Do you really want to have a hyperactive grandparent living in your family? I don't know. We need to think about that carefully. But it's interesting that these individuals seem to have survived unscathed for many, many years despite this. And so what we think is happening here is that IGSF1 is a critical regulator for TRH signaling through the TRH receptor. And so when you have an IGSF1 deficiency, you have defective signaling through both of those routes, meaning that you end up with a lowering of both TSH and also prolactin. We've just frozen our slides again, I think. And what I was going to go on to say is that we also have interesting data, which I'm not going to share today, showing that GH is also potentially crucially regulated in GH and IGF-1, not IGSF1, but IGF-1. And so we know that GH signaling is not quite right in these individuals as well, and there are some issues around growth retardation in children, but interestingly, some of the adults have the opposite phenotype, almost an acromegaloid phenotype. Okay, here we go. So looking at the pattern of hormone deficits and the response can help us to identify which gene to screen. Is it important to do this? Yes, it's crucially important, because if you don't identify these individuals with congenital hypothyroidism early on in life, you can end up with severe lifetime sequelae. And can you make a difference if you do make that diagnosis early? Yes, you can. Here, for example, are 4 children that we've been associated with, and you can see the age at diagnosis as early as 5 weeks, 8 weeks, or 14 weeks. If you're at 14 weeks, it's too late. You finish up with major neurocognitive deficits. At 8 and 5 weeks, you're doing better, but you've got some deficits. But here's a child picked up because it's an individual in the same kindred, so diagnosed at birth, replaced immediately, completely normal development. So making these diagnoses is really, really important for affecting the long-term outcome with these children. I'm afraid we seem to be hitting the same problem again. Okay, so moving on, I'm now just going to talk briefly about the fact that until a few years ago, we considered thyroid hormone uptake into cells to be some sort of slightly odd passive process. The thyroid hormone just arrived at the cell surface and made its way across. We now know, because of the sorts of individuals that I'm about to describe to you, that that's not the case, and that thyroid hormone uptake into the cells is very tightly regulated through transporter proteins such as Monocarboxylate transporter 8. And here's a child who was referred with major neurodevelopmental issues and hypotonia, which is a key feature of this disorder, with central hypothyroidism apparently, T4 low, TSH not as high as one would expect, Although, interestingly, look at the circulating free T3 level. This is actually raised. So this is a very peculiar biochemical picture. Apparent hypothyroidism, but with a high free T3. And these individuals cannot take T3 up, so they have a specific defect in monocarboxylate transporter 8, the transport protein which is responsible for getting T3 into the brain. And this has taught us an awful lot from the study of these individuals about how T3 is handled in the central nervous system. So we know that T4 and T3 need to cross the blood-brain barrier, and they do so by specific transporter proteins. Once inside the CNS, T4 can be converted into T3 in the astrocytes, but for T3 to actually find its way into the neurons to do its job, or into the supporting oligodendrocytes, it needs to be able to transfer through the MCT8 protein. And if you have defective MCT8 signaling, then you do not have T3 action in your neurons, and you finish up with a major neurodevelopment condition. So we now understand more about how T3 is transported. I also mentioned, of course, that T3 is the active hormone. T4 does nothing. So even if you can get T4 inside the cell, you have to convert it to T3, and that is done by a family of proteins known as the deiodinases. And the deiodinases are a perfect example of molecular control and regulation. So T3, the only hormone that does anything, is generated either because T4 is converted into T3 or because T3 itself is directly taken up. But how do you protect the cell against excessive thyroid hormone activity? Well, there are enzymes, other deiodinases such as D3, which can inactivate the T3, converting it to T2, or turning T4 into reverse T3. And these are again inactive. And again, studying individuals with very unusual patterns of thyroid function tests has helped to shed light on the role of these proteins. So here is a 36-year-old gentleman who was referred to us. Goodness knows why he had his thyroid function test checked, because he had Raynaud's phenomena. And yet he had a very high FT4, but with a normal TSH and a normal 3T3. And he looks as if somebody who is failing to convert T4 to T3. So you would hypothesize that he would have a mutation in one of the deiodinases, either DIO1 or DIO2. But try as I might, I could not find any variation in his deiodinase gene. But then we remembered, and working on and building on the work of Sam Refetoff in Chicago, that the deiodinases are part of an unusual family of proteins known as the selena proteins, and that's because they contain that unique amino acid selenocysteine. And this individual had a low plasma selenium level, and plasma selenium is dependent on transport around by a selena protein. And so this led us to hypothesize that these individuals would have a defect in the formation of selena proteins. And to remind you how that happens, the specific tRNA that carries the signal for the selenocysteine to be incorporated into the protein is only anchored when other important transcription factors are available, and in particular this entity here known as SBP2, which is the CeCH BP2 protein, and this has to be functioning normally for selena protein formation to occur. And the long and short of it is that these individuals have defects in this, and therefore they don't really make properly any of the selenocysteine-containing proteins, of which there are 25 important ones in humans, and you'll recognize these because these are crucially involved in antioxidant properties. So they're protecting us from oxidant stress. And what's interesting is this world of looking at thyroid function tests gets you into studying all sorts of other areas which you don't have great expertise in, because you start looking at all the things that antioxidants are involved in. So it turned out that our gentleman had complete azoospermia with spermatogenic arrest, and that's because he does not have normal sperm maturation processes, and that's likely due to the fact that the antioxidants, glutathione peroxidase, thyrodoxine reductase, and others, are not expressed within the testis of this individual compared to normal subjects. This individual had a very strange posture, and he had a muscular dystrophy phenotype, which is very similar to individuals who have specific mutations in just one selena protein causing a myopathy. And this leads to the sepin myopathies, and our patient had that phenotype as well, almost certainly because of a more global problem with selena protein production, and photosensitivity. Of course, antioxidants protect us against stress, and when we did a UV test on this individual, we had to curtail it because he was particularly sensitive to UV light. What's an interesting phenotype now coming to light in these subjects, which we haven't previously reported, is that they appear to be getting quite marked and pronounced cerebral atrophy, and they also are developing quite marked aortic disease as well. So studying simply an individual with an abnormal set of thyroid function tests has led to the recognition of an entirely new human condition of SBP2, or selenoprotein binding protein 2 insufficiency, which has many, many different features, all recognizable because of impairment of selenoprotein function, but easily diagnosed because of a very, very distinctive thyroid signature because of a lack of deiodinase activity, high T4, normal T3, normal TSH, a new set point in the axis. So we're nearly there. We've taken our journey all the way from production of thyroid hormones under the regulation of TSH, its transport into the cells, its conversion to T3. Now it has to get into the nucleus. Now it has to bind and interact with its protein. And this is where I started my PhD work in Chris Chatterjee's lab more than 20 years ago, looking at thyroid receptors. There are two main thyroid hormone receptors, TR alpha and TR beta. Each has a splice variant encoded off different chromosomes. And the body has thyroid receptors in pretty much every single tissue, but what's interesting is the balance of those receptors differs in the different tissues. So the beta receptor dominates in the axis centrally. It is the isoform which is critically responsible for negative feedback regulation of TSH and indeed T4 and T3 production. It's also the dominant form in the liver, whereas the alpha isoform is present in the heart, the CNS, the gut, and the musculoskeletal system. RTH beta, thyroid hormone resistance beta, was first described by Sam Refetoff, 1967. So this is a very important year for us. It's 1 in 40,000 live births as we hit the anniversary, and there are many, many individuals around the world affected by this with many, many different mutations. Try as hard as we did, it took us until 2012 before we found the phenotype within the human TR alpha receptor and the description of RTH alpha. And these are two very, very, very different conditions. So mutations in different isoforms of the receptor producing different diseases. RTH beta will be well known to many people, and I won't spend much time describing it. Many of the individuals with this only come to light because their thyroid function tests are abnormal. Many of them appear to be relatively unaffected apart from having a goiter because they've got continuous TSH drive. In essence, the impaired negative feedback of T3 at the hypothalamic and pituitary level means that the axis is set to a new place where you need more T4 and more T3 in order to drive TSH production. So you finish up with hyperthyroxanemia with non-suppressed TSH. That is the diagnostic hallmark of this condition. It's important to make that diagnosis because the majority of these individuals do not need any treatment. The last thing they need is antithyroid treatment for inappropriately diagnosed hypothyroidism. They don't have that. They have a compensated state because they have resistance in their tissues. RTH alpha is a very, very different condition. So here, the problem is defective signaling in the brain, the heart, the gut, and the musculoskeletal system. And this was the very first human to be recognized with an RTH alpha mutation that we reported back in 2012. It was a young girl who was referred to us because of growth disturbance, and it was a very unusual form of growth disturbance. She was of short stature, but this was not within the upper part of the body. It was very, very dramatically restricted to the lower part of the body, the lower segment. So the subischal leg length was markedly down compared to her, for example, sitting height. There are some other very important, interesting features. These individuals have macrocephaly, very large heads. They get many skeletal manifestations, delayed fusion of the fontanelles. Some of these features look like classical hypothyroidism that we rarely see nowadays, Wormian bones, delayed eruption. a very dramatically delayed bone age if you compare their absolute measurements compared to their chronological age. Gastrointestinal manifestations, terrible constipation. This poor child was only able to open her bowels about once a week at best, and actually was also referred to one of my colleagues in parallel because of the concern that she may actually have childhood obesity. This was no childhood obesity. This was major distension because of malfunctioning of the colon. And if you do colonic manometry in these individuals and compare it to the normal spikes in contractility, there's virtually nothing going on at all in these individuals. You note that they have a very, very distinctive biochemical phenotype. They have a T4 level, which tends to be at the low end of normal, but a T3, which tends to be high. And this results in a very strange T4 to T3 ratio. And this is a more difficult biochemical signature to recognize, but it exists. So the T4-T3 ratio is low compared with compatriots. And that ratio is one of the big fingerprint clues if you see a child with apparent hypothyroid phenotype, but what look like relatively unremarkable TFTs. The adult phenotype exists. We've now seen several adults with it. They have a very interesting similar phenotype. Here we again see the short lower segment. Here we see the macrocephaly again, the large head, and also some interesting skin changes. And actually the phenotype varies. The first individuals that we saw carried the more severe mutations in RTHα. There were significant issues around cognitive performance, but now we have kindreds such as this, where we have individuals who have mutations which are less dramatic and which are therefore associated with a less dramatic phenotype. You still see the macrocephaly, you still see the abnormal TFTs, but these are individuals who are working normally in society. One of them has been to university. So not all of this phenotype is doom and gloom. So there are many, many emerging mutations in RTHα, and my prediction is that there'll be just as many of these as there are RTH-beta mutations. It's just that we're some 45 years later coming to the game of recognizing these. But the features are very distinguishable because they're typical of individuals who've got defective signaling in the alpha receptor tissues. So what I hope I've shown you today is that actually studying people with unusual thyroid function tests actually helps our understanding of the axis more widely and helps us to understand many of the strategies and approaches that we use to treating more common forms of thyroid disease because we understand the mechanisms through which thyroid hormone is acting and being regulated. So next time you're sitting in your clinic and you see somebody with unusual thyroid function tests, don't ignore it. If you do nothing else, get on your email and send me a note or give me a call because we might be interested in it. You never know what the next clue will be to understanding how this quite amazing axis works. So please take away this summary diagram of what these disorders can look like and how they can present. Here are my acknowledgements for all the collaborators that I work closely with, and I particularly like to point out my long-term mentor, Chris Chatterjee. And as I say, if you have any queries or any questions, then feel free to contact us. We're very happy to advise, and I already have collaboration with many people here in India, and actually there are lots of interesting new findings coming out. Thank you very much for your attention.